So we're going to go ahead and take notes, and we're going to take notes on let's start with the first quarter. So this is the final exam, what's going to be on it, what do you have to be able to do. And then once we kind of have an outline of the final exam, I'd like to start reviewing by looking at the new study guide that I've given you. Even though it's a new study guide, it's going over some of the old stuff in the first, I want to say, one half of that packet. There's some activities I want you to do in class as a review for your exam. So we started out the year, the one of the first topics we discussed was what? Do you remember? Besides how to take notes, besides the 6 R's, what were it? What say that? DNA? Yeah, that was one of the. It was a little later though. Before that, we talked about the elements, right? And we talked about specifically elements of life, and we talked about, and you can go back and watch videos if you want, but the schnapps, right? The carbon, the hydrogen, the nitrogen, the oxygen, the phosphorus, and the sulfur. And we said that these are all in various elements, and it was really key, I told you, that if you learn these, and you learn which one goes where, when you're looking at some really complicated molecule like DNA or RNA or a protein, you're going to have a good idea of what it is. And I said because we talked about biomolecules next, right? Besides chemical, we talked about chemical reactions, right? We actually did a couple labs with chemical reactions. We talked about physical reactions, the difference between physical properties and physical changes, right, versus chemical reactions. We talked about things like concentration, right, that we saw really started to become important later on. Concentration, solute, solvent, solution. Does everybody agree that we've talked about all this? We've done all this? Yeah, okay. So we know that all life, we call ourselves, life on this planet is called the carbon-based life form. We're carbon-based life forms because almost every biomolecule we have, every biomolecule, of course these are the elements, and then we talked about biomolecules. And yeah, over there, or somewhere around, yeah, there. Are you guys done with the recycling?
Yeah. They just want to go with other containers away.
Wow, you guys are really fast. All right, so biomolecules, they're also known as, and they're a type of macromolecule. And macromolecule is big molecule of life. And remember, you guys were freaked out because you didn't know what a molecule was, but now we do know. It's a bunch of elements. A molecule are elements that share electrons, right? And a chemical reaction is when you rearrange those sharings. So I think we're pretty good with that. I think we have it. We've seen it. We've applied it. We're carbon-based life forms. So now, what are the biomolecules that we've talked about? Now, I want to emphasize something here. I'm going to say clearly here, and I need you to really, really think about this. This is really a summary. All right. So you really need to think about that. You need to know the details. I can't go back over 2 months, 3 months worth of work in a day, correct? So what I'm doing is I'm reminding you of what is going to be on this test. You have notes, you have study guides that you filled out, You have notes that I've sent you in your Jupyter from other students who've taken good notes. In fact, if there's anyone in here who has all that was willing to get some extra credit by sharing, we'll go and scan it during fourth period. I can go scan it if you leave me your notes. That would be very useful, so I can just email the whole ninth grade a copy, one set of notes. So at least if you haven't been taking good notes, then everybody can have a copy. But you have your notes, your old quizzes, your old tests. I can't go back and print all those tests and quizzes, so if you don't have copies of them, or the discussions of them, or the overviews, you have your YouTube videos of all the day's lessons, all posted online. So you have all that as a resource. Of course, don't forget, you have Khan Academy, right? You have each other. You have study groups. You can start forming study groups now. Would really be a smart, especially if you share more than one class with each other. You don't have to be friends, guys. Study groups, friendship actually sometimes can be a hurtful thing in a study group. But if you get a couple people that know what they're talking about, and you're quizzing each other and working out problems together, it turns out to be very successful. All right, so molecules. What are the biomolecules that we talked about?
Nucleic acid.
That's right, very good. So we talked about nucleic acids. It's good that you mentioned, great. All right, so I'm going to have to switch to my finger, and it never turns out pretty. So it would be helpful if you don't laugh at me now or later. All right, so, yeah. So we did talk about nucleic acids. And what elements are involved in nucleic acid? First, what types of nucleic acids are there? The two that we talked about. DNA, RNA. And what are the elements that, and you know what, why don't we do a Venn diagram? Because we did one of those in our last lab reports, right? Why don't we go ahead and do a Venn diagram? We'll have two circles. And one will say DNA, and the other will say RNA. And what do they both have in common? They both have the same elements, right? What are those elements that both DNA and RNA have? Carbon is number one. Carbon's going to be number one in just about every biomolecule, right? In fact, every biomolecule. Phosphate is correct. Phosphate backbone. What else? Nitrogen, right? Because they're nitrogenous bases. Oxygen, correct. These are the elements in RNA and DNA. What other elements would there be? Is there another element in? I'll pause here and let you look at your notes.
Hydrogen. They said hydrogen.
Hydrogen, correct.
Ain't that it?
Is that it?
Yeah.
Is there sulfur?
No.
No sulfur in nucleic acids. Where is sulfur usually found?
Protein.
Correct. See, we learned something. Good. Proteins is correct. So, RNA. What's the difference? All right. So, what else do they both have? We know they both have all these elements. That's good. What else do they both have? Say what? Okay, nucleotides. They're both made of nucleotides. There's no D. That's right. The three that they both share. A, which is at A, T. Let me make that neater. That's right. And G. That's right. Guanine, thymine, and adenosine. Right. Those three. A is adenosine. Huh? Oh, yeah, I don't even know what I'm thinking. It's not, you're right, it's not T. It's A, what? C and G. All right, cytosine. All right. We're all good? All right, so those are the three. What else do they share in like their structure, DNA and RNA? Right, they have that's good. They have a 5' end in humans and a 3' end. The 3' end has a hydroxyl group, right? The 5' end has a phosphate group. Remember, phosphate is phosphorus with 4 oxygens, but we draw it like this. Okay. They both have a phosphate backbone. What does that mean?
It's held together by.
That's right. It's held together. Each strand is held together by a phosphate backbone. By phosphate connected to oxygen, connected to carbon, connected. There's a phosphate connecting each of the what? The nucleotides. Each of these nucleotides are connected together by phosphate. Okay? That's what that means. Does anybody have problems with this?
No.
Okay. Don't say no, because somebody in here might, and then they're just going to be embarrassed because they don't want to say it. If you have a problem with this, and you don't want to say it out loud, I need you to write it down and somehow get it to me, or a Jupiter grade message me, or somehow privately give me information that if you're embarrassed for some reason you shouldn't be, but that's fine, and I will address it tomorrow. Is that clear? So you have to actually, here's where that Cornell Notes it makes a big impact, right here. If you're not writing down questions that you have, you can't ask them, now or later. If you're not writing down your thinking, you're going to forget your thought. Does that make sense?
Yes.
So write it down. When I don't see a notebook open, I'm concerned. So a phosphate backbone, 3' nucleotides. What else? All right, so that's it. So that's DNA and RNA. There's something else that they both do. They both do something called hydrogen bonding. Now, the hydrogen bonding in RNA is different from that in DNA. What is, and we'll talk about why in a minute, but they're both hydrogen bonding. In RNA, it just creates a shape. In DNA, it holds two things together. What two things does it hold together in DNA that makes it different? Two strands. So DNA is double-stranded. And these two strands, how are they connected? They are what? They're not parallel. They're anti-parallel. What does that mean? That means that the 5', that means the 5' on one strand is attached to the 3' on the other. All right? That's anti-parallel, it's double-stranded, okay. Hydrogen bonds hold them together. What else does DNA have that RNA doesn't?
DNA is missing a O.
Right, excellent, very good. It's a deoxyribose. Look at all this that you know already. You feeling more confident that you're answering these questions? Yes? Deoxyribose. I can't stand using my finger. I hope the pencil hurries up and charges. It's so much neater with a pencil. It's out of charge.
Swear to God.
Deoxyribose sugar. Remember, that means that it's missing oxygen, right?
At the second problem.
That's right. What else does DNA have that RNA doesn't?
DNA has T and R.
That's right. That's perfect. There's a T. Excellent. Nucleotide. If you know all that, you're pretty solid with the difference between DNA and RNA. This macromolecule we can almost check off. RNA is the what? Single-stranded. It's pretty much a lot of opposites. There's no antiparallel because there's only one strand. Right? So that's the thing. The hydrogen bonds impact its shape. That's going to be really important in the next unit because we're going to be talking about protein synthesis. We're going to be talking about something called tRNA. And tRNA is an RNA that actually has this. I'm just going to give you a little bit of foreshadowing. tRNA actually turns out to look something like this.
I like a bird.
Yeah, it's a horrible drawing. You know, I can't draw.
Yeah.
Let's just face facts. I try. I'm going to keep trying. I'm not giving up, but there it is. So it looks like a T, okay? It's not why they call it a T. That's not why they call it T because it's a transfer. It transfers amino acids. Is why it's called a T. But it looks like a T to me. So that shape though is because of hydrogen bonding. So RNA has shape. In fact, ribosomes. You know that organelle ribosomes that we talk about. It turns out ribosomes, don't do that. Let me pause. It turns out ribosomes are also made of RNA as well as some proteins. So RNA does a lot. It's not just informational. DNA is pretty much informational. By the way, that's the purpose of DNA is information. RNA is used for information. But it's also used as, it can also be an enzyme, all right? And it could, I hate it could be an enzyme as well. And it could actually be used to transfer, it actually has other functions as well. So RNA is much more versatile. In fact, it was thought for a long time that RNA was the most versatile, the most interesting macromolecule is actually a protein. They're the most complicated. So it was for a long time we thought proteins were where the book of life would be, right? There are 20 different amino acids, correct? 21 different amino, right? So if there's 21 different amino acids and there's only 4 different nucleotides, which do you think you should be writing a book with? The one with 21 letters or the one with 4 letters? If you're talking about writing a book as complicated as as a human being. 21, right? So most people thought a protein had the best shot of it, but it turns out nature actually picks the simplest answer. It turns out that it's that DNA that is the source of our code. So anyways, DNA and RNA, those are the big differences. Of course, there's U, right? U instead of T. That's it. If you know that, you're going to pretty much know the difference between DNA and RNA. Now, of course, with the bond, with DNA, let's go ahead and just kind of do this. With DNA, you got A matches with T and G matches with C. I think that's pretty straightforward. When RNA when RNA, RNA doesn't have a second strand, but RNA does match with DNA during during RNA transcription, right? And later, RNA transcription. Also, it happens in ribosomes as well. What you have, A is not going to match with T, it's going to match with U, and G is going to match with C. So there is this pattern, this matching that you have to understand. Are we all good with this? All right. So that's DNA. That's nucleic acids. That took a long time for us to understand. I hope you do. You do. Now let's go ahead and talk about another one. Give me another one. All right, lipids. What are lipids? Fatty acids are an example of lipids. So they're long. What are they? Long chains of what? All right, hydrocarbons. They're long chains of carbons, hydrocarbons. So they're long chains of carbon and hydrogen, right? So there's carbon, 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 and then there'll be hydrogens on it. You okay? Wow, that's kind of harsh. All right, so anyways, long chains of carbon and hydrogen, there are going to be some oxygens involved, but what do you know about the oxygen rate relative to carbon and hydrogen? It's going to be a small amount. Only a very small amount of oxygen. Some lipids, depending how complicated, because you can have glycolipids, which have sugar on them. You can have all kinds of combinations of stuff. So some lipids, like phospholipids, have phosphate, right? So there are other elements that are involved in lipids as well. So phosphorus could be involved in it, but for the most part, it's carbon, hydrogen, a little oxygen, maybe a little phosphorus. Those are lipids. So the types of lipids, and some people have already mentioned them, there's fats. There's a really important one that we talked about a lot, is phospholipids. And then there's another big one that we call steroids. And the steroids, the steroid that we really focused on is a big one called cholesterol. And cholesterol is in the membrane. It has a function. The function of cholesterol is to keep the membrane fluid. And of course, the membrane is made of phospholipids. All right, different phospholipids make up different membranes, but that's fine. And these fats are used for storage of energy. Also, fats can be used for insulation. They can help keep you warm. And of course, there's that physical trauma protection, right? A person is larger, it's harder to do damage to their organs and to the sensitive areas of their body. The thinner you are, the more easily you can be damaged, right? As far as being physically hit by a car or whatever. A larger person has a better chance of being protected, their organs protected by fat. Fat actually does act like a cushion. It protects against pushing. Push. No? Okay. I was just trying to rhyme, see if you guys can remember it. So it's hard, it's harder to hurt someone who's bigger because of that. All right. And a larger person is warmer in the cold in the wintertime. As someone who's fat, I can tell you I'm a lot warmer than skinny people are outside. I'm always seeing little skinny guys just like, and I'm like eating my apple, just relaxing. I'm like, it's nice out, and they're freezing. Now the opposite happens in the summer though. The skinny people are like, dude, this is nice, and I'm like, God, where's the air conditioning? I need water and air conditioning and ice. So yeah, there's insulation is a real thing. Fat does insulate. It does protect you against heat loss. All right, so that's lipids. What's another biomolecule that we talked about? Proteins, correct. In fact, we're going to be talking a lot about proteins next.
Never mind, I'm sorry.
You doing it?
I don't know.
Proteins. Proteins are made of, what elements make up proteins? Carbon, sulfur, I heard. Hydrogen. I hear a lot of talking, ladies. Hydrogen, sulfur, carbon.
I think nitrogen, that's it.
Nitrogen, that's correct. For the most part, that's it. Carbon, hydrogen, nitrogen, oxygen. Oh, there's oxygen as well. There's a little oxygen there, right? Who remembers what the thing is called that puts it? By the way, these are all. There's only one of these that's not a polymer, right? Nucleic acids are polymers. Are lipids a polymer?
No.
Not a polymer. Not a polymer. Proteins, on the other hand, are polymers, are they not?
Yeah. Amino acids.
If you have a polymer, you have to have a monomer. The monomer is amino acids. Who remembers what the structure of amino acids are?
What you mean, like the shape, like how it look?
The shape is correct. C double bond O OH, the carboxyl group, that's on one end. It's connected to what?
H.
Another carbon. Then what? How many bonds per carbon?
4.
4. So
Another carbon.
so there's 1, 2, 3, 4. This one's done. Every oxygen has 2. There's 1. It's connected to hydrogen. Here's another that's connected to carbon. So that's 2 for oxygen. And here's 2 for this oxygen. So these are all done with their bonds, right? This carbon is connected to what? Stop having conversations right in front of me. You're not looking at the board, so you can't possibly. It's connected to an R group. The R group, how many different R groups are there? 21 different R groups. And that could be, then the other side's connected to a what? An H, a hydrogen, and then it's connected to a what? Is it an O?
No, it's missing.
Close. Look it up. Look in your notes.
I don't even remember this. Oh,
First of all, your notebook's not open. I don't even know if you have a
I
notebook.
don't
'cause
have my notebook.
Oh, I see.
It's in my
Everyone else, here's my question. This could be a test. I can make an easy test and give you a good grade right now or a bad grade if I just ask this question. Pull open your notes to the page that has the amino acid structure. We covered for a week, so you should have it in your notes. Written in your notes. If you don't have it, your notebook, you have an F on your notebook grade. If you have it, you have an A. See how easy I can grade your notebooks? Everybody should be able, and if you're finding a hard time, let me say something very quickly. I'm trying to teach you something here, right? So don't think that I'm upset, because I know you're ninth graders, I know you're still working on this, but look, if you can't quickly find something you need, if you're going through your pocket, the pocket of your binder, and you're trying to figure out where it is, then you are not organized enough. Your notebook should be organized by date. There should be little tabs on it. Say, oh, here's proteins, here's lipids, here's nucleic acids. Yeah. It's an amine group. What does it look like? The carboxyl group is here. So the carboxyl group is here.
That's the carboxyl
Right, so here's an N, then what?
Yeah, I got that right here. It's in my notes. PBS, but still.
If you, and here's my problem, you're gonna fail your notebook grade. This is, again, your notebook's gonna get graded next week. You're gonna fail your notebook grade, because you shouldn't have PBS stuff in your biology notebook.
It's not in there, it's not PBS thing. I'm
You
sorry,
should
I just
have,
put
and
PBS.
if you don't have this content in your notes right now, in your biology notes, I'm just warning you, I'm not trying to frustrate you. You're not gonna get a good grade on your notebook, because we covered this a lot. It's demonstrating you haven't been doing your work. And it should be in your binder, in the 3-ring binder, in order by date or by topic, however you particularly ordered it. But there should be Cornell notes, specifically from the date that we had assigned as a class. We all agreed to a date that we would start this assessment on. So don't be surprised if that happens. I don't want you getting upset later. So proteins. So this is all they're all connected to each other, you see. And this is your monomer, and you just constructed the monomer of a protein, amino acid. There's 21 different R groups. Some of these are acidic, some of these are basic, some of these are aromatic, some of these are long chains, some of these are really short, they're all different. That's going to be important when we look at DNA and something called transcription, because that's our next unit. So that's going to be important when we start making these proteins, when we start looking at genes, because what do genes do? What do genes do? All right, that's it. Really. Here's what genes do, and this should be an answer. It should always be the first thing that comes to your mind. Genes make proteins. When I look out among you, and I see people with their back to me, especially those people that haven't turned work in, and I see glowy screens on their glowy light on their faces, I got problems. We're in the dark. My God, especially in the dark, I see the light of your phone on. All right. So proteins are polymers of amino acids. They're polymers of amino acids. And so we can connect them together. What do they do? What do proteins do? They're the functional unit. They do everything. They function. They have primary structure. What's their primary structure determined by? What are they determined by? What would the answer? If this was the question on the test, what would you put in the blank? That's an interesting thing. You say R group. The R group, which amino acid goes where is the primary structure, right? So if you have 3 of the same amino acids in a row connected together, and then you have a 4th different one, a 5th is the same as the first 3, that's your primary structure. That is the primary structure. But what determines that primary structure? So if I have an alanine, these are different amino acids, right? Guanine, cytosine. If I have these in a row, this is first, this is second, this is third, this is fourth, and this, of course, can go on for a long time. These proteins can be very long. This structure, this order, the order of the amino acids is the primary structure. That's what primary structure is. So what determines the difference between guanine and cytosine is their R groups. They have different R groups. That's it. Because this part, everything else you see here, every amino acid is exactly the same. It's this side chain, this R group that sticks off the side, is different from each. These two are the same, have the same R group. They're exactly the same, right? Because they're both A's. The G has a different R group. The C has a different R group. Is that making sense to you? And you can have repeating ones, or you can have different ones. All depends on something. What determines what amino acid goes where in this order? When you're putting them together, when your ribosomes put together these amino acids, what is going to determine that order of amino acids? No, these are peptide bonds. They're holding them together. What determines it? Okay, someone said something. What did they say?
Somebody said a chain.
A what?
Somebody said like a chain.
A chain. No.
A gene.
A gene is correct.
We said that the first thing.
So the gene determines the amino acid sequence. I will pause this, rewind it, and let you listen to pure silence.
I heard.
So if you whispered it,
and I copied
if
over.
you whispered it and I didn't hear it, do you want me to stop this and rewind it?
Yeah, because Layla said it and then I copied off her. I was like, yeah.
You might have thought if you didn't have confidence, I'm sorry, but it is a gene, is correct. All right, enough. So we're talking about a gene here. That's what a gene does. A gene makes a protein, and which protein it makes is going to determine whether that protein, how well that protein works, what does it do, and we'll talk about that in just a second. What's the fourth biomolecule? Carbohydrates. Carbohydrates, the most probably the most important one for all life on Earth, most life on Earth, is glucose. Glucose, and we talked about it a lot. It's six-sided. It's a hexose. It's six-sided sugar. But of course, there's another really important one that we talked about, and that's ribose. And there's two forms of ribose. There's the deoxy form, and there's the regular ribose, and that's a pentose sugar. That's five-sided and 5 carbons. So 5 carbons with an oxygen there in the center there. So each one, remember, each connecting point is a carbon. So that's a ribose. That's 5 sugars. You might remember fructose, which is also 5-sided, even though it's still a hexose. It's still 6 carbons. It's just it has 5 sides, but that's all. So those are kind of the monomers. These are the monomers of carbohydrates. There's a few more. There's galactose. There's sucrose is a disaccharide. So, but those are the monomers. When we put them together, we can make some really big polymers, and one of them is called starch. And we saw starch granules in cells, plant cells, right? Starch. And those are for plants. And what does starch do? It stores energy. Correct. Very good. And what is another? What else do they do? What other kind of? We talked about in animals is something called glycogen. And of course, we talked about cellulose. Cellulose is again plants, but it's not for energy, because even plants don't use cellulose for energy. Even plants don't break it down for energies. Cellulose is for what?
Outside covering.
Okay, that's fair, but it's for structure. It's a structure. Good thinking though. I like your thought, right? Because you're thinking bark, right? You're thinking the woody part of that's true. But it's the structure of the plant, the cell wall, right?
Which one is used to like?
Now, that's how we connect the cells to this biomolecule stuff. The cell walls that you looked at that you're studying for your cell part of your test, the cell walls that you're studying for your cell part of your test, they're made of cellulose. All right? Glycogen in humans is found in the liver. And of course, that's for storing sugars as well.
Isn't that like bile in the liver?
Say again?
Isn't that like bile in the liver?
It's a good question. Bile is not in the liver. It's produced by the gallbladder, by the I believe it's the gallbladder. The bile is used to emulsify fats. So when you eat fat, you have to make it, you know, fat doesn't mix with water. We all know that. Well, bile helps that fat become more soluble in water, so it emulsifies it. All right, this is what we call emulsification. 2 minutes, very good, thank you. That's very helpful. I wish more people would do that instead of complain after. It's really helpful instead of complaining.
Clearly.
Wait, somebody else, what?
Whenever I tell you to do that, you be like, well, we're still learning. You told me to stop packing up.
No, I didn't, did she start packing up?
I mean, she
No, she just said, oh, we have 2 minutes left. I said, thank you. She was polite. She was polite. She wasn't interrupting,
oh my God.
right? She's not packing her stuff up like, I'm ready to go. No. She's just like, we have 2 more minutes left.
It's one now.
It's one now.
Well, because of you.
Don't, you childish, Mendoza.
Why are you saying childish? See, now you're being interrupted. I'm at the right, yeah.
No, you don't.
All right. So genes, just quickly, here's the thing. We're gonna talk about this tomorrow. Gene gets coded to mRNA that goes to a protein. That's what we're gonna talk. It's called transcription. That is the goal for you to understand transcription and how proteins, they are what determine a trait. They are the trait. They are the product. It's called the gene product. Is this comfortable? Is this something that's made you comfortable with the content? All right. So let's make sure. There's a couple little things that left off here. The glucose, remember, is CHO. And remember, it's C. The general formula is for every carbon you have 2 hydrogens and you have 1 oxygen. So if you have C6, so think about it. If you have C6, how many H's do you have? 12. And how many oxygens?
6.
So this is why glucose is C6H12O6. If you have C3, then you have C3H6O3. See?
Because of 1 to 2 to 1.
It's a 1 to 2 to 1 ratio. So this is the ratio here. That rate is 1 to 2 to 1. All right. So that way, that's just to help you identify this stuff. Obviously, I'm. Okay. Now that that's all over with, let's move on. So carbohydrates, proteins, lipids, nucleotides, nucleic acids. Don't forget, I put nucleic acids here, but really, well, I'm going to post this so you don't have to worry about it. This shouldn't be nucleic acid. It should be nucleotides, right?
On the lipids part, after physical, what does that say?
Where do you see that?
On the lipids.
Physical trauma protection.
Oh.
So it protects you against physical trauma. All right. So that's pretty much all the biomolecule stuff that we talked about. And we're going to talk a little, but remember, we also, after that, let's go on to the next page then. After that, the second mistake I made is it's not 21 amino acids, it's 20.
Wait, where are you back now?
I hate when I do that. I do that often. I apologize. I don't know why I'm constantly thinking 21 amino acids is
Wait, Mr. Where
20.
you making mistake with the multiple?
And we started to put together those molecules into something called a lipid membrane.
Oh, you never heard of
Is this new? So lipid membranes is new to you?
Oh, all right.
Is it?
No, I really don't remember talking about lipids.
Me neither.
I really don't.
Oh, really?
Nobody else does.
The membrane, the membrane, the phospholipid membrane.
Oh, never mind.
Oh.
You never called it that. Right, you never used that term.
Oh, I see.
said phospholipid.
Oh, I see.
Yeah.
I see.
Right. Now apologize to all of us.
Right.
I'm waiting.
All right, I'm moving on. So remember, it's also known as a bilayer.
Oh, that's what it. I really hope Kimberly.
It's phospholipid.
She gonna get you.
What is it? Phospholipid bilayer. All right. So phospholipid bilayer, remember, is just these phospholipids all interact, and we talked about things. Let's see what we talked about. We talked about things like hydrophobic, and hydrophobic, remember, is non-polar. Hydrophilic can be polar or ionic, right? And we talked about this membrane as being a separator, as something that separates the outside from the inside. We said, make sure that you know that this is three-dimensional. We're only drawing it in 2 dimensions, but you know, it would really be more of a sphere, a hollow, an inside, that where the outside is being separated from the inside by this membrane. And of course, like we said, all things that you're going to separate, if you're going to have something that's alive, that's going to you need to be able to get things in and out, you're going to have a protein, and there's several what we call integral membrane proteins. Integral, meaning a part of, membrane proteins. And we said that this protein has to have different sections. You have to have a kind of a hydrophilic part that sticks out of the cell, and a hydrophilic part that sticks inside the cell. And then you have to have kind of a hydrophobic part that sticks with the tails, right? That's what I say. And of course, we also said that we have these things that are called cholesterol. It's a kind of a ring structure that sits in the membrane, and cholesterol helps it stay fluid.
Didn't you say it was a certain way the tails had to go or something?
Yeah, the tails go to the face each other, and then the heads they go to the intracellular and extracellular.
Exactly. Perfectly said. Well said. So the heads are pointing towards the extracellular portion, which is outside the cell. So let's go ahead and draw it. Hey,
It's okay, Mendo.
you want them green? You want me to make them green?
We can't see.
Oh, the green is maybe you gotta get glasses or move closer to the screen.
Y'all see me? I still can't see it. I'm right here, Mendoza.
I can see. I'm right next to
You
you.
got the bifocals on.
Then it sounds like you all need glasses. I do have bifocals. She's right.
I know. We know. Think about thick.
Is them what's they called when they turn into sunglasses when you go outside?
Yes.
My brother got them. My little brother got them.
These are everything I can afford. They're Armani.
Period. He said Armani.
That sounds expensive.
Not really. I mean, they are, but not really. All right, so cholesterol helps the membrane stay fluid. Now, why is this important? Because as it gets cold outside, you don't want your cells start to freeze and break, right? It gets cold out there. I mean, think about a fish swimming in the North Atlantic, right? Very, very cold water. You don't need your cells freezing and breaking apart. So in animals use cholesterol to keep this membrane from sticking together and freezing into a solid. So it keeps the membrane fluid. In fact, that's why we call this model, the model of a cell membrane, the fluid.
Mosaic model.
That's right, Mosaic model. So why is it called the fluid mosaic model? It's called the fluid mosaic model because it's made of a bunch of different components and it's always moving. It's always moving and changing. And it has to change. And the example that we keep using over and over and over again, and you can expect to see it on the test, I can guarantee it since I'm writing the test, or at least finding the questions, is when you have a membrane, and this time I'm going to draw it like this, you're going to have receptors. They're outside the membrane. And these receptors are going to be able to pick up signals that are going to tell the cell to change. There's a lot of conversations going on that worry me, especially since I'm going over the test. So if we draw a cell isn't just phospholipid bilayer with little doors and windows in it. They have, they have little channels and carriers in the membrane. That's true. All that's true. But they also have these little things called receptors that stick out. And these receptors that stick out, they're usually glycoproteins. Again, if you go back and look at your old study guide, you'll remember glycoproteins. The glycoproteins are proteins that are attached to sugars. All right. Whatever they look like, these glycoproteins are they make these binding units, and part of that glycoprotein crosses the membrane. Part of it crosses the membrane here and here, and then part of it might even stick inside the membrane. So the hydrophilic parts stick inside, and outside hydrophobic sticks in the membrane. And then what happens is that you have these molecules that may get produced by some other organ in your body. So what would this, does anybody remember what this example that we talked about over and over and over again in class?
No.
Insulin, correct.
You what? You just did you? The question gonna be.
I did just do it
What is
yesterday.
the fluid mosaic?
Oh, is that what I said?
We don't know.
So the question gonna be, what is the fluid mosaic model?
The question's gonna be whatever comes into my head when I make the test. No, it's not that.
Try to pee. I want to see it. That's God.
Okay, you're losing focus, ladies and gentlemen. It's 1:03. We really need to finish this, so try to listen, all right? You did amazing today, amazing. Thank you. But and you're answering questions, you're staying focused. Hey, let me be very clear. I have appreciated your performance today, but let's not lose it the last few minutes. Now, this is called insulin, right? Insulin is at another cell in the pancreas, right? So in a cell in the pancreas, this is the pancreas up here. In the pancreas, there's these cells that make insulin. They do exocytosis.
How do you spell pancreas?
How do you spell pancreas?
Yeah.
Pancreas.
Pancreas.
Forget
Pancreas.
the A that he said before the E.
Yeah.
It's
At this point,
Pancreas.
All right, so exocytosis, out comes from that pancreas, out comes that insulin molecule. That insulin molecule is known as what? What do we call the insulin? Hormone is correct. That is a hormone. That's the example of a hormone that we've used over and over and over again. Insulin is a hormone. Hormones are used to signal. I'm trying to connect these ideas of proteins and lipids and carbohydrates together into this thing that we've been studying now for the last 3 weeks, cells. right? So exocytosis, the pancreas cell releases this insulin. It goes through the blood to the various cells of your body. They match up. They pair up with these receptors, and when they pair up, they bond. And what happens is that the outside then signals the inside, hey, release a channel. It says the message is release the channels.
The message is release the channel.
That is what the message is. Release the channels. And what happens is that then the cell receives that message, and the vesicle with all the proteins that starts to release these channels. The channels then get embedded in the membrane. And what do these channels do?
They release.
They're in the membrane now. They've been released because of insulin. What are they going to do? What are they going to allow in? Sugar. So sugar now, specifically glucose, is going to go into the cell through the channel. And it's leaving the blood. So now you lower the blood sugar concentration because the sugar is going into the cell. Then the cell can start digesting the sugar, breaking it down, making energy, making ATP, storing it as whatever it's going to do with the sugar. But to get the sugar out of the blood, you need that insulin molecule, that signal. The cell, this pancreas, this organ called the pancreas, it has receptors that measures the concentration of sugar in the cells. Does that make sense to everybody? I don't know why this thing's not working anymore. I don't know what's going on. All right. So how much time do we have left in class?
None.
Like
5 minutes.
All right, 5 minutes. None, girl. All right. So the pancreas measures, then decides whether to release. Remember, that's exocytosis. And you have to know the steps of exocytosis if you want to be ready for this exam.
Well, you said this right here is
The release of the insulin by the pancreas cell, exocytosis.
Wait, say that one more time.
Exocytosis. Exocytosis. The release of anything. Anything that the cell puts out, it's it's exiting.
so exocytosis.
So cytosis. Cytosis is from cyto means cell. So anything exiting the cell is called exocytosis. So when you start drooling, what are your cells doing? They're releasing mucus. They're releasing mucus.
Why people drool in their sleep?
Right.
We'll talk about that some other time. So so when you drool, you're releasing mucus. Your cells are doing exocytosis. When your nose starts to run, your cells are releasing mucus. They're doing exocytosis. Stuff that's inside the cell, that's being stored in the cell in vesicles, is released. That's what these pancreatic cells do. They release the hormone. That hormone is a signal. It's saying, hey, yo, cells, we got too much sugar in the blood. Put the channels in your membrane to get the sugar out. When the blood sugar gets too low, the cells in the pancreas stop making insulin and start making glucagon. The glucagon's another message. Hey, get rid of the channels. Stop taking the sugar out of the blood. Yo, liver cells, release the glycogen. Break the glycogen down and release the sugar. We need sugar now. So your body's constantly sending these messages. It's called cell signaling. That's how your body works. That's how hormones work. And every different action, a young lady or a woman swelling up during her period, getting bloated is a reaction, is a response to a hormone that's being released.
Okay.
Yeah.
Okay, so since exocytosis is the release of something from the cell, endocytosis is the intake of something?
Yes, correct. So endocytosis is the taking something into the cell. So like what? What would you have to take in, if you have a cell, what would you have to take into the cell? Well, sugar, insulin. Well, insulin never actually enters the cell. Water, but those things can move through osmosis or through a channel. Though it's still endocytosis, the key is endocytosis are things like food, right? You're right, all that stuff is true, but don't forget food. What about a white blood cell? Here's this bacteria. You get infected by a staphylococcus. And staphylococcus, you know, it's a staph infection. It is horrible. It's a staph infection. It could kill you. All right. Staph infections can be deadly. So you have a staph infection. What is your body going to do? Is it just going to die? No, it's gonna fight it off. What's it gonna do? What it's gonna do is gonna have an immune response, and it's gonna release its warriors, and the warriors are white blood cells.
That's where you get pimples from, right? The dead warriors.
That's right, partly.
Not the dead warriors.
Okay, you have to pay attention. We're running out of time. So what's gonna happen is that this bacteria is going to get surrounded, the white blood cell is going to surround it, and what's it going to, hey, stop with the giggling now.
It's gonna take, then it's gonna die.
It's going to, well, not right away, it'll get overwhelmed eventually, but it's going to surround the bacteria, it's going to surround it, and eventually, what'll happen,
It's gonna eat it, close it.
that's right, it's going to close up, you got it. It's going to close up, and inside it's going to have the bacteria is going to be trapped in a membrane, and then it's going to release the lysosomes. This is a test of your understanding. There's a vesicle in your cell called lysosome that's going to bond to it, and it's going to release enzymes. It's going to kill it and digest it. That's what white blood cells do, or at least some.
They be fighting in you.
Peroxisomes are similar.
Yes. The difference between lysosome
Yeah.
and peroxisome.
Like, one of them act as like a garden sugar, and then the other one does something.
What do white blood cells do again?
They are part of the immune response. They destroy invaders. It's important that when we think about exocytosis and endocytosis, we think about all the processes that involve things leaving the cell and going into the bloodstream, right? So what is a list of those things that would do that? And we've already talked about like the pancreas releasing the release of hormones. And we've talked about the things like the release of mucus as just two of the many different things that have to leave a cell in the form of exocytosis. Of course, every cell has to get rid of waste. If you think about it,
What happens if they don't?
they die as waste. You know, what if you don't poop? You would die. You would build up all that poop, and bad things would happen. Endocytosis, then we have to consider what are the things that have to go into a cell? Well, obviously food, and in the case of white blood cells, any kind of invaders, right? Of course, water. In the one form of endocytosis is this thing called pinocytosis. So that's the water going in large quantities, and then of course we have phagocytosis, which we looked at white blood cells as an example. So at this point, we should know what exocytosis and endocytosis is. Remember, as you saw with white blood cells, when you're talking about phagocytosis, or you're talking about major changes in structure, in cellular structure. Major changes, ladies, major changes in cellular structure. So that means that when that white blood cell is moving in between the cells, in between the tissues, you have to take that that cytoskeleton, that those things that are made of the filaments and both the micro and the intermediate, right? The micro and intermediate filaments that you should know what they are and what they're involved in. Both the micro filaments and the intermediate filaments, as well as the tubules, microtubules. Those are three organelles, three structural parts of the organelles that you have in your study guide that you're expected to know for the exam. So think about it, when that white blood cell moves around your tissues to go and hunt down those bacterial invaders, that white blood cell has to change its endoskeleton, has to change the membrane, has to reform itself. So you saw how quickly it did that. So this is all very fast and very complicated. But so just the idea that when that cell, if we think about this as being kind of a close-up of what's going on inside the cell, the organelles, the nucleus is sitting in here in the cytoskeleton, the gliolis, the Golgi bodies, all kind of the ribosomes, the mitochondria, they're all suspended in this matrix, this made of filaments and intermediate filaments and microfilaments and tubules, and they're all getting ready to do whatever it is that needs to get done to move that white blood cell in between cells to get to the bacteria. So when we talk about endocytosis and exocytosis, a lot of this is going on then too, because as you remember from the previous part of this video, the cell has to actually move itself around the invader or the food particle or whatever and surround the object, in this case it's the red dot, the object in a membrane that allows you to fuse. If it's a piece of food, it's a food vacuole. If it's an invader, it's a bacteria in that vacuole and that vesicle, and you have to fuse it with a lysosome or peroxisome that's going to just tear apart whatever it is so that the cell can use those resources and get rid of the invader or whatever the case may be. So that's exocytosis and endocytosis. So, of course, movement across the membrane is dependent on one major concept, really. What is it dependent on? Normal. Let's say we have two kinds, right? We have passive and we have active, correct? Two kinds, passive and active. When we're looking at, why don't we do that Venn diagram again? Because I think I always like that. I like this idea of a Venn diagram. You hate those? What would you rather have? Would you rather have a chart?
Yeah, I just don't have enough room. That's why I'm like writing those.
It's easier to write a chart in the notes.
Yeah. But I mean.
Okay, so let's do passive, what's active, and what's both.
Yep, that's what you say.
All right. Whatever works for you. So in passive transport, bless you, what kinds of things go in passive transport? Osmosis is passive. And what else? Say again? I can't hear you. Facilitated diffusion. Can I put diffusion in general first, just because it'll make more... Is that all right with you? I mean, it's your answer. All right, so we have diffusion, and then we can have, of diffusion, we can have either simple diffusion, and we can have facilitated diffusion. Is that all right? Everybody okay with that? All right.
Why you writing so spaced out?
Because this is what you should do when you're taking notes. It's a good question. It's a really good question. Yes, you can add things in case you get right. You should always leave space in your notes. So if there's changes to be made, or you read something you want to add to it, or you have some questions later, you should always leave enough space to do that. In this case, I know I'm going to add some stuff, so I made sure I left space. So simple diffusion is based on what? Like what's going to be? It's going to be high to low, right? That's a big part of simple diffusion. And in simple diffusion, what is it that moves? What kinds of things move with simple diffusion? Say what? That means that, let's think about what simple diffusion means. That means there's no facilitation, no protein is necessary. You just have the membrane and the things go right through the membrane. What kinds of things go right through the membrane? I can't hear you. Solvent? Sugar. No, sugar wouldn't. Not salt. Salt don't, no.
She didn't say that.
No, she was in, someone else said salt. Someone else said sugar.
I said solvent.
Solvent, okay. No. So the kinds of things that move through simple diffusion, again, please review whatever you need to review. As you're moving through this review that we're doing here in class, this outline, as you're identifying, and that's part of your grade for next week when I grade your notebooks, I'm looking to see your reflection. Where is your writing? When you're taking notes, where is it that you're saying, I need to study this? I need to go find a resource for that. I don't understand this. Where are your questions? Because I hear you, I'm paying attention. I see that you now have some of the detail. That's fine. That's why this is called a review. Then next week, when I look at your notebooks, I better see those questions there. I better see some references, some evidence that you've gone and quote unquote studied and found those answers that or at least have the questions to ask the questions in class. So things that move high to low through the membrane without any help would be small and nonpolar. Maybe polar as well, depending how small they are, how strong the concentration gradient is. So small and nonpolar are the things that easily move across the membrane. What are some examples of small and nonpolar objects or molecules? There would be things like carbon dioxide, CO2, oxygen gas. So these are gases. Gases easily move across the membrane because they're nonpolar, they're small. So that's simple diffusion. Now salt and sugar's big and it's polar. Salt is ionic, so it's fully charged. That's not going to move across the simple diffusion through a membrane. That at best you're going to get is facilitated diffusion. And facilitated diffusion is with help of proteins. With the help of proteins. What kinds of things move with the help of proteins? Anything, really. Anything that's obviously some things are so big that they have to actually be engulfed through phagocytosis, right? But if things are going to move across the membrane with that could be anything. Well, what kinds of objects? Of course, you're still going from high to low. Why are we still going from high to low? Because it's passive. That's what passive means, right? That means if you're going from high to low, you don't need any extra energy. And the example we keep using over and over again that you should have clearly in your mind is if you're dumping water and it's moving downhill, you don't need anything to help it. It'll move all by itself. Gravity will just let it flow downhill. However, if you're trying to get water to go from low to high, you need to have that. You need to carry it uphill, right? So let's think about that and keep that in mind. So with the help of proteins, you're still going from high to low, so everything's good. And so what kinds of proteins are involved in this? Do you guys remember the kinds of proteins? That's right. Carrier and, channels are two examples. Oh, sorry. It's hard to write and think at the same time. Channel proteins and carrier proteins, right? What are the differences between carrier proteins and channel proteins? Well, carrier proteins, they generally are going to have kind of a shape that's going to allow the cell to change the protein to change its shape. So if you came in and this molecule came in and looked like this, it would fit in there, and let me erase it. And next week we'll look at the example of sodium potassium pumps as a good example of a carrier protein. But something looks like that, and over time, if this is outside obviously, and then what the protein will actually do is it'll change shape and it'll move like this. So now that charged particle or polar particle is now inside. Does that make sense? Literally takes it and brings it inside. That's a carrier protein. A channel protein is really much easier. It's just this idea, and then we've drawn this about 100 times, that you have just a hole in the membrane that's created specifically for a molecule. And remembering that in both cases, you have the hydrophilic portion of the protein on the outside and the inside, and then you have that hydrophobic portion that's in the middle, that's in the membrane. So that would be more of a channel protein. So those are facilitated. Osmosis is just simply the movement of water, the diffusion of water. So again, from what? From high to low. So in all these passive forms, it's all high to low. It's all with the concentration gradient. Now, diffusion can get some help. Osmosis can get some help. Remember that there's kind of a link. There's these things called aquaporins.
Called what?
Aquaporins. They're literally channels that allow water to come in. However, water doesn't need an aquaporin. Water can get through the membranes without aquaporins. It's small enough that it can fit in between the lipid molecules. It finagles its way in. All right, so active. So that's all passive. What's active diffusion? What's active transport then? What kinds of things are we talking about here? All right, pumps, right? Very good. And what do pumps move? What are we going to move if we're doing a pump? Right, pumps are going to move things. All these active are going to go from low to high. In fact, we can put high to low here for passive and just emphasize low to high for all the active ones. Also, we can even put the use of ATP, right? So usually active is going to require either ATP or some kind of energy, and low to high. So pumps are a really good example, and ion pumps, specifically ion pumps. And we're going to look at sodium potassium pumps. We might even get a chance to do it today if everything goes smoothly. I do. You got the bar. I want it back. It's my favorite pencil. That's the second favorite pencil. I'll give you my second favorite.
Wow, thank you.
You have to click it.
I just got confused. Okay.
All right, so ion pumps move from low to high. They require energy. All active transport requires energy, so any kind of carrier or if anything is moving from low to high. What is another example that we just got done talking about of something that's actively transporting across the membrane? That's right, exocytosis and endocytosis. That's a big change. I mean, you can imagine all that changing that we saw the white blood cell do when it phagocytosed. Remember, endocytosis is both pino and phagocytosis. So all that movement of the cell that we saw white blood cells do takes a lot of energy. I mean, you're breaking apart proteins, you're building them back up. I mean, there's just a lot of ATP used in that. So your body, your cells use a lot of energy to do this endocytosis and this exocytosis. But for sure, it's going to be active transport. So what do they have in common? They have movement across the membrane. And that's pretty much all they have in common. I mean, if you think about it, one requires energy, the other one doesn't. They're both moving across the membrane or changing the membrane to get the substance in or out of the cell. However, they look very different if you pay attention to the details, right? They're both dependent on a concentration gradient in different ways, but still. And one requires energy, the other one doesn't require any extra energy. So is that reasonable? All right. So let's go on and do some practice problems, some actual movement, some osmosis problems that you're going to see. What are you going to see on the test that looks like this? You'll get some questions, you'll see some scenarios, I'll make some drawings, I'll say this scientist looked at this membrane and they saw this thing happening. What kind of transport is this? What would you call that? So you'll see, I'm going to put an asterisk by them. Yeah.
Depending on what?
Depending on concentration. So I'm gonna put a little dot, a little asterisk by the things that are definitely going to be on the test. So you're going to see exocytosis and endocytosis on there, and pinocytosis, pumps. You'll see diffusion of water. You'll see simple diffusion, and you'll see carrier proteins and channel proteins.
So everything we wrote basically.
Here on this page, yeah. This is very important. This is very important. So it's important to me that you know it, because you're gonna get into course questions on this as well. So yeah, definitely know these not just their definitions, but if you were given, ask yourself something. If you're given an example, concentration. Let me write the whole word out. I see that's a common. So when you're taking notes, you have to get used to being able to write abbreviations, right? Instead of having to write the word concentration out. It's horrible for your practice of your spelling, but it's really good to quickly write down ideas and get them in your head long term. So concentration, depending on concentration. So if I give you a scenario, if I give you a drawing of a membrane with whatever the scenario, any one of these scenarios, could you identify it as being simple diffusion, or as carriers doing facilitated diffusion, or as channels doing facilitated diffusion, or could you identify it as endocytosis, exocytosis, or ion pumps? If you can identify them, if you can look and say, can you answer the question, does it require energy or not? So those are the kind, you could, I know you could. Let me give you an example. So, I mean, I'm sure some of you won't be able to, but that has to be a kind of a personal choice at this point. So you have a membrane. I draw it something like this, sketch it out, maybe it'd be fancy. This is your membrane, and then you have this thing drawn out like this, and then it would be labeled. It'll probably you'll have it labeled something like this outside. Maybe it'll say extracellular. And this would be inside. That's right, intracellular. And I put a bunch of material out here. Maybe there's a number, maybe it's like 20, but maybe there's no number. Let's just use an example with no number. And then I put some of it in here. And I say, The movement is, now watch the distance. Watch how easily I can change the question for you. If I say, this is the movement of the particles, what are we talking about? Who can describe it? Yeah. Facilitate diffusion. How are things moving? From high to low. Facilitate diffusion. No extra energy required. Those three things right there I knew just by a picture. I didn't even have to have any words. Things are moving from, just an arrow. Things are moving from high to low. So I know it's passive. I know it requires no extra energy. I know there's a channel protein, so I know it's facilitated. Done. Easy. These are easy questions if you know what you're talking about. But of course, you know, the reality is that that's true of anything, right? Things are easy if you know what you're talking about. Things are always hard if you don't, but it's all about whether you're prepared or not, really. Want someone to close the door, please? I don't know what's going on out there, but I'm really just trying to focus. So if I draw something like this, though, and I take this and I draw the arrow in this direction, what are we talking about here? Active. Why? Because this is low and this is high. How do I know that's low, that's high? My God.
Started from the bottom.
Well, not because it's on the bottom, because I could read it.
The air's going up instead of going down.
Well, okay, don't make that mistake. So you're right, it is going up. You're right, and that part's not wrong. What it makes this active is that how many of these little dots are there on this side? What makes this low is not that it's at the bottom, because there's barely any of them here. If I count them 1, 2, 3, 4, 5 of them, where here there's 20 or 30. That's what makes them higher and low. So in other words, that's a good point. I end up making that mistake, and I keep giving you examples where the high is up here and the low is down there, and I think that's a really clear misunderstanding that could be facilitated by my lack of detail here. So let's go ahead and do this. If I do the opposite, if I do this, right? And there's just a few of them here, and I go and I draw the arrow now in this direction. Don't say it's going from high to low, right? Because it's easy to make that mistake, because this is up and this is down. Well, that's true. On the board, this is up, this is down. But it's really low concentration to high concentration, right? All right, so don't make that. It's easy to make that mistake, so it's good. I'm glad you mentioned it, all right? So you want to go, if you see it going from low to high, you need extra energy. Excellent. I love it when somebody learns something. That's fantastic. Thank you. Excellent. So you're going from low to high, then you need extra energy. And what kind of active transport is this? Is it a carrier? Probably a pump, right? Some kind of pump. And by the way, if I'm drawing it accurately, I'd have to draw this. I'd have to say ATP being going to ADP. That's the use of energy. This is the example. This is the use of energy. So I'm using energy to move things from low to high. Again, the example I keep saying, I hope you listen at some point. If I'm moving water uphill, I have to carry it uphill. It's not going to move by itself. So I need to put energy into that. The energy of the cell. The energy of the cell. And I can promise you that'll be a question on the test. The energy of the cell is at ATP. ATP is how the cell does anything. The example I used in class repeatedly throughout the semester, the quarter, is look, if you're going to buy a house, you're not going to pay somebody in cookies. Nobody's going to give you the keys to a house, the deed to a house, because you made them, a million cookies. That's not reasonable. The only thing that buys you a house in this society is American dollars. Agreed?
Yes.
Okay, you gotta, try to focus here. In a cell, the only thing that gets things done, the only thing that gets any work done is this thing called ATP. So make sure you know that, because if you're gonna see, if you see anything happening where you see ATP going to ADP, then you're using energy, so what is it? Active. All right. Yeah. All right. I went over it in class yesterday, but let me go ahead and draw it out again, because I didn't put it on the slide. But ATP is adenosine. That's that A that's in DNA and RNA. I see there's a lot of people still having conversations, and it's really getting to be annoying. I've been trying to have patience. I'm asking you to try to stay focused. You have that adenosine, and then you have 3 phosphates. Now remember, phosphates, phosphorus with 4 oxygens. They're all attached to each other. You know, like the phosphate in the backbone of DNA and RNA, right?
Yeah.
So these 3 attached to each other. That is ATP. There's a lot of energy in these bonds. Remember we talked about bonds having a lot of energy? There's a lot of energy in that bond. This releases the most amount of energy. This releases a little more energy. And this one releases the least amount of energy. But there's energy here to be used, they exchanged, that can be used to run other things. So that's ATP. ADP, this is called, again, adenosine triphosphate. Adenosine with 3 phosphates. You can have, as you go from ATP to ADP, this is ADP, adenosine what? Diphosphate, right? Just 2 instead of 3. When you did that, when you lost that phosphate, that energy was released. And you're able to move, in this case, move things from low to high. So when you see this happening, ATP going to ADP, that energy is being released so that you can make that change in that protein. That ATP going to ADP. By the way, this is kind of an aside. Adenosine is the stuff that's in DNA and RNA, right? Is that is A the only nucleotide that's available? No. So we could actually have GTP and CTP and TTP and even UTP. So these are possible, but this is really the one we see a lot. So this is one you're going to really need to know. But just know that you can have 3 phosphates on any one of these, and they can be used to release that energy, because it's this bond between the last 2 phosphates here, that's where all the energy is coming from. And you can either put, and by the way, you can reverse this. Let's be clear on this, because when we look at a mitochondria, what a mitochondria is going to do, all right, girls, I'm going to have to ask some of you to leave, because you're not cooperating anyway, so being here is not helping you, and you're also going to be distracting, so that can't be. So if I have ADP, again, that's adenosine with 2 phosphates, and I put energy into it, right? I put energy into it. To it plus an extra phosphate, what do I make? ATP. So they're reversible. I can make ATP, put energy into the molecule, or I can use the energy and make ADP. So they go back and forth. That's the currency. That's how you exchange energy. You put that energy in the molecule, and when we get into cellular respiration next, we come back into January, we're going to look at cellular respiration, photosynthesis. You're going to see that that's how we do it. That's how plants do it. They take sunlight and they put it into ATP. And then they make sugar. They put that ATP energy into sugar. Then we take that sugar and we take the energy out of sugar and we put it into ATP. So we do all we do, everything goes back to exchanging. The middleman is ATP. The thing that's going to make it, bless you, the thing's going to make a difference is this ATP. All right. So that's kind of the energy and the movement across. Now let's look at some problems and some examples, and then we'll move on. I'll see if anybody has any questions. So if I give you a cell, and it could be something like this, right? Or it could be a U-tube. Remember the U-tube that we looked at earlier? I could have a membrane. Either way, you have some kind of selectively permeable membrane. And I can give you numbers, or I can just put dots. I can put 1, 2, 3, 4, and I can put a ton of dots out here. And there's always the question, you always ask that, I mean, my God, you have to ask this question. If you're not asking this question, it's likely you're gonna get it wrong. What question do I have to ask before I start answering which direction anything's moving? Right, where's the lowest to high? That's a good question to ask. Where is the highest concentration? Outside is the highest, so high is here, and then solute, high solute out here, right? And here's low solute. So let's be clear on that. We've identified that. So good question, we've answered that. What else do you have to answer if you're going to ask? Because right now I could say the ions are going to move from high to low, and I could be wrong. What do I need to know before I can answer that? Yeah. That's jumping the gun. So the question is what? Is it going to what? Is the solute going to? Can the solute pass? So is the membrane permeable? Is the membrane permeable to the solute? In other words, does the solute cross the membrane? That's the question you have to ask yourself. That's it. If the solute can cross the membrane, in which direction will the solute move without any problem? If the solute, high to low, which would mean if you were asked to identify it, it would be the arrow would look like this, correct? If the solute can move, it'll go from high to low, go from out to in. Correct? Yeah. Okay, so easily, I can change the question really easily. All I gotta do, and by the way, if I'm telling you this, what am I likely gonna do? I'm likely gonna change the question this way. If I just say it doesn't cross the membrane, if the answer is no, now what's gonna happen? The solute can't go from high to low, what's gonna have to happen? It can't cross, the solute can't cross. So what's going to happen? It can't move. The solute cannot cross the membrane. You're close. There's someone said in the back, listen to each other. What's going to happen? If the solute cannot move, what happens? Well, the solute is not going to move, so it's going. But what is going to move? Water is going to move.
Yeah, you said that.
In which direction? So there's like, remember I told you there's like two different ways to look at it. You can say from where water's its high to its low, or towards where the solute's higher concentration. Either, I don't care how you guys look at it. It really doesn't bother me. So water's going to move out. That's it. That's it. That's right. Water's going to move out, and just like someone just said a minute ago, I don't want to mention you because I'm recording, it's going to shrink. Right? Just like you said. Why is it going to shrink? Because water's moving out. Does that make sense? Yeah. So I can ask you any number of questions. I can draw any number. I can draw like 1, 2, 3, 4. I can just write numbers, or I can put little dots. I could put 10 molar, 3 molar. I could put 3 molar, 10 molar. Or you know what? Just to make it easier and not get away from molar, why don't we use percentages? That's easy to understand. Yeah. So 10%. 50%, 20%, 20%, 30%, 10%, 5%, 2%. So again, two questions. I can change them up really quickly. Does the solute move across the membrane or not? So you got A and then B can have different scenarios. How quickly I can write these questions. I love these questions. They're easy to grade and easy to answer, easy to write. In what direction, if I say the solute cannot cross the membrane, in which direction would I draw the arrow for what's going to move in what direction for A? If the solute cannot pass.
That's where the solute
So the percentages are solute concentration, correct. So is solute cannot pass, so in which direction is water going to move?
You wanna go inside.
Water is going to move, someone says inside.
It's gonna move out.
It's going to move out because it's salty, it's higher concentration of solute.
Oh, I thought
So,
it was solid.
yeah, it's solute, so it's moving out. So good, it's good. So you got to make sure, that's a good point. You have to make sure, concentration of what? In this case, concentration is solute, all right? So always good questions to ask. So if we say the solute can pass, is it can pass now? In what direction is the solute going to move? You say in,
It's gonna stay.
it's going to stay the same.
In and out.
In and out, good. It's going to move in both directions at equal rates. Done. Isotonic, yeah. See, 20% is isotonic. These two are so funny. You two are funny. You and McCann. Yeah, I can't say it all. It's hilarious. You bother each other so much. I know, I see it. All right, so see.
Didn't nobody say in and out.
All right, okay, all right. So let's look at C. We got 30% inside, 10% outside. Now let's go ahead. What do you want to make it? You want to make it pass or not pass? Does the solute move or not?
Yeah.
All right, yes, I'm moving. I'm going to say yes or no. Let's say yes. Solute moves. What direction is solute going to go? Out. Done. And I'm putting red for the solute so you can see, right? The solute's red. So it's easy. Now let's go ahead and say the solute no. D is no. The solute can't move.
That's like 2%.
Yeah, this says 2% and the other's 5%.
It'll go in.
So what's going to go in, what's going to move, and in what direction?
Water goes in.
You're going to say water's going to go in because that's where the solute's the highest concentration. That is correct. So water's going to move, water's going to move, solute's going to move, and solute's going to move.
So,
That's it, guys. That's all there is to it. Real simple. Boom, boom, boom. Easy. Either the solute's going to move or the water's going to move. If the water moves, it's osmosis. There's no extra energy required. The other possibility is I could give you a question like this. We have different, you have the equal levels. I say this is 5%, this is 10%, and I say to you, the solute does not pass. No. What's going to happen? Water will. In which direction? To the right or left? To the right. And the water's getting, so this water level is going to go up. So instead of there, it'll be up here. New level. That's it. It's all based on some really simple ideas. Things are going to move from their high to their low unless they can't, and if they can't, then the water will move. That's that simple. And the opposite direction.
Well, how do you know when they can't move?
How do you what now?
How do you know when they can't move? How do you know when they are?
How do you know when they can't move across the membrane? Somebody has to tell you. Like that has to be part of the problem. You can't just memorize it. However, there are some tricks. I've seen the state of Ohio do a couple interesting tricks to this problem. And it has to do with what you just asked. Did it just do that? Again. Okay, so, there's an interesting problem. There's an interesting problem that the state did to 1 year in their practice exams. They said, look, what if I had the cell, oh man, they don't tell you if it's going to pass or not. They just say, here's a cell, and sodium ion is 30% out here and 10% in here. What's going to happen? If they tell you it's a cell and they tell you sodium ion, it's ionized, so they're testing two things. Do you know that ionic particles are not going to cross the membrane without help? the one thing they did tell the students was that there is no protein to help, right? No facilitated. So if there's no protein, and this is just a lipid bilayer, and you have 30% sodium ion and 10% sodium ion inside, they're not going to tell you if it passes or not. Guess what they want you to figure out? Does it pass? And would it pass? No, because it's charged. So what's going to move? Water, and it's going to move in what direction? Out. Beautiful. So it's going to be like 5 questions on the test. You should be able to get 5 questions really easy just by being able to do that. Fair?
Yes.
So like 5 or 10 questions about like different carriers. Is it passive? Is it active? And then 5 questions that, where you're actually applying it. Osmosis, I saw that. Osmosis and diffusion. But no, you're perfect. You don't do anything wrong. But I'm just making fun because of our previous conversations.
I think it looks like it's taking a picture when you do that.
It does, doesn't it? It freaks me out. I agree. I've thought that more than once. Yeah. Yeah, right past. All right, so we've gone over passive and active transport. We've gone over the cell structures, the different ways things move across the membrane. You have to know all the different cell structures. Anybody have any questions at this point?
You go back to the other.
Are you posting the video?
Yes.
You go all the way back to the first.
You want to do what now? Go back where? I don't understand. You two are ridiculous. I don't understand. Yeah. What a great question. Why is ATP more common than the others? It's probably has something to do with evolution. Probably has something to do that the original cells had used ATP, and then that's what's used in all these different processes. But honestly, I don't know the answer why ATP is more common than GTP, UTP. So, good question. Yeah.
Okay, so you know how you said it could be GTP, CTP, all that? Can it be GDP and stuff like that?
Yeah. There's the DPs. That's what's going to happen with all the TPs. They're going to become DPs. Yeah.
And can it be like other unique things, like how the T and the D? Can it be other ones?
It's those. It can be anything because this is chemistry, really. But it's those 5: A, T, G, C, and U, because that's what's available for the cell.
No, I'm saying can it be different, like the G, the T and the D, like di and tri? Can it be like this?
Say what?
Tetra?
Oh, could there be more? Could it be more than tri? No, it gets to be too energetic. That once you get past 3 phosphates, it becomes too unstable. I'm sure it happens, but it would quickly become triphosphate, because any quad or hexophosphate would just be way too energetic. It would just fall apart. Question? It's a great question, though.
So ATP comes from the mitochondria, right?
Yeah.
Where does ADP come from? Or is that just like
It's a good question. Let's get into that. But first, actually, what I had planned to do, I would love. See, this is always where I'm kind of at a fork in the road. Because for me, what I want to do is I want to get into mitochondria. Like, I'm really excited. I want to talk about energy. I want to talk about mitochondria and chloroplasts and get into how do we make sugar and how do we break it down. I love, this is like one of my favorite sections here. However, I also love the other road, which is talking about genes and proteins and how we make those, right? So transcription, translation, and it's an application of the organelles and that's what the unit I already photocopied and gave you. So I'm going to leave that question that you're asking for January, all right? And we'll talk about mitochondria and chloroplasts, which is a great question. I'm very excited to answer it, but I'd like to focus first on genes and protein synthesis first. Yeah.
So you go back to the beginning when you had exocytosis with release of hormones,
The cell signaling, what do you want to know about that?
I was saying if you could go back, I missed one of the parts.
Can I just post it tonight and then you can look at it over the weekend and fill it in? because I'd like, if you don't mind, I'd like to kind of go into, I see you're getting it from the notes, so that's fine. All right. So, you know, this is that fork in the road. I'm looking at mitochondria and chloroplast, and there's something interesting, but I'm going to let it go. We'll get back to it.
Extra credit, like after school hours, you know, like a quick lesson on it.
If you'd like, we can, you know, next week. But we're going to do it. It's just it's about when should we cover it, then, now or then. But let's go ahead and pull out your new study guides. Remember the new study guides. The first 2/3 of it are review. Oh, we only got 6 minutes. It's still good to look at it. Could you get me one of those new study guides in the back? There's a big pile.
You should have one. I'm prepared. I'm taking points off.
All right, you can take points off me. Oh yeah, you don't grade me. Boom. Thank you very much.
Listen and take some money off your check.
All right, we got DNA. You can take me off so I don't go on the next extended trip. Thank the Lord Jesus. All right, DNA and RNA.
So don't do that.
I did have fun actually, so I guess I would be losing out. So DNA and RNA. Guys, the first half of this, first 2/3 of this, we're going to do this next week. The first 2/3 of this new study guide is actually review.
Are we supposed to be able to see these words? 'Cause I can't see them.
Okay, focusing on not wasting time. Let's move on. If you can't see, I don't know why. I'm older than you.
This, not this.
Oh, yeah, I don't know what that is. Those are not words, dear. They're letters. So let's start off with this word that everyone should know at this point. The genome.
Genotype.
Like genotype, right? Like, it's where genotype comes from. The genome, an organism's genome, you have a genome. The genome is all the DNA for that species or that organism. New study guide, RD to page 88. The title of the page is The Genome. Very first page, inner page. What you see there in front of you that someone said, are we supposed to be reading that word? That's not a word.
I just looked
That's
at
a-
that real quick.
It is. It is a sequence. It is a sequence. It is a sequence of nucleotides. It is DNA, and what you see there is A, T, G. If we are going to draw it out, it would be something like this, and right next to it would be hydrogen bonded to it. Would be hydrogen bonded to a T, A, C, G, A. So understand, when you're looking at this, what they're just looking at the sequence of one strand. They assume that you understand, if you know this sequence, you know this sequence, right? Because they're opposites, they're base pairing. If you got A, you know the other side's a T, etc. So there is the sequence of DNA for this virus. They picked a virus because a virus is very simple. It's small, it's simple, it's not a cell, it's not a living creature. It is just simply as some DNA inside a package. In the case of the gamma, I think this is gamma anyways. In the case of this, gosh darn it. In the case of this gamma X174, it's a bacteriophage, it's a virus, huh? Guys, there's a lot of mumbling going on, it's distracting.
X174.
Yeah. Please look at your thing. As you look at gamma X174, they draw it out like this. That DNA is that virus's genome. That's the genome for the entire virus. It's not very big. We have genes that have bigger sequences than this. Our DNA is billions long, 3 billion, actually 2,500,000,000, but whatever.
Of course you know.
But
We both said that.
there's this particular virus has a genome that's about anywhere from 500 to 1,000 base pairs. And you see it listed there. And yeah, it's tiny. You don't need to know all the details. But what is interesting is that even a virus, and you'll notice that the questions they ask you are, can you convert, bases to kilobases, et cetera, et cetera. I'm not concerned about that. Please do it. It's good practice for you. You need to be able to do this. So you should be able to convert all the questions on that page. You should be able to answer without any help from me. But what is important that you understand is this idea that there's a beginning, there's a gene in this genome. There are genes that start with ATG. All right, we'll begin. Thank you. Do you have a pencil for the rest of the day?